adolescence, different cortical regions maximally develop and mature at different ages. Elucidating the temporal sequence of human neurodevelopment, or in other words, the spatial progression of developmental plasticity across the cortical landscape, has some important implications for understanding mechanisms underlying this plasticity in health and disease, the optimal timing of youth interventions, and as I'll highlight at the end of the talk today, the developmental origins of adult brain organization. Nonetheless, the precise spatiotemporal sequence of human neurodevelopment has yet to be really systematically characterized. Shown with human neuroimaging for large-scale features such as cortical thickness, myelination, and connectivity, and by animal model studies characterizing the maturation of plasticity regulating biological features such as cardinal beam and interneurons. But critically, beyond this coarse division, there is additional temporal variability, but a parsimonious framework that accounts for this variability is still being established. So in a recent review article, our lab described one unifying framework, which posits that during childhood and adolescence, cortical maturation progresses along a sensorimotor-association axis or gradient.
Sorry, we're having some Zoom issues. I'm giving 2 seconds, I'm sorry. How are we going on Zoom again? Once got eyes on it. Is that all? Excellent. Great. Yep, all good. So to recap, in a recent review article, our lab was proposing this idea that during childhood and adolescence, cortical maturation may progress along the sensorimotor-association axis or gradient. So the sensorimotor-association axis, or the SA axis, as I'll call it, is a gradient of brain organization that is globally arranged from primary cortices at the sensorimotor end to modality-selective and multimodal cortices in the middle, and then to transmodal cortices at the association end. And this axis is often called the unimodal transmodal gradient after Daniel's pioneering work. And also it looks a lot like the gradient fish that Claude showed us, so I love that. But this axis captures continuous and concerted variation in remarkably diverse features across the cortical sheet, including but not limited to some of the features shown here, like cortical connectivity, cerebral blood flow, gene expression, laminar architecture, task-based activation, and evolutionary expansion. And the SA axis that I'm showing on the left is actually just an average of the 10 maps shown on the right. And as you can see, this sort of representative SA axis captures both graded feature changes across all 10 maps, but also at the same time reliably captures divergence between sensorimotor and transmodal association cortices across all 10 maps. So we hypothesized that this axis may capture not only the spatial layout of cortical properties, but also the patterning of developmental plasticity. And in order to test whether developmental plasticity may be governed by the SA axis, we studied age-dependent changes in spontaneous cortical activity. Spontaneous or intrinsically generated activity plays really vital roles in the developing brain, influencing, for example, neural migration, circuit wiring, sensory maps, synapse refinement, and myelination. Moreover, studies of the developing murine sensory cortex have repeatedly shown that as the cortex transitions from plastic to mature, spontaneous activity evolves from strong and more globally synchronized, as depicted on the left, to sparser and decorrelated, as shown on the right. So we aim to non-invasively study this sort of stereotyped refinement of spontaneous activity in humans using resting state functional MRI. And as we all know here, fMRI is certainly not a direct measure of neural activity, but many studies employing simultaneous fMRI and electrophysiology or calcium recordings
have shown that a greater level of spontaneous activity and more synchronized activity will increase the amplitude of low frequency bold fluctuations, suggesting that bold fluctuation amplitude may be higher when cortical plasticity is enhanced. We therefore predicted that as plasticity is reduced and the cortex matures during childhood and adolescence, bold fluctuation amplitude would exhibit sort of heterochronous declines along the cortex's SA axis. We tested this prediction in a cross-sectional sample of 1,033 youth, ages 8 to 23 years old from the Philadelphia neurodevelopmental cohort. And in the sample of participants, we quantified cortical bold fluctuation amplitude by calculating the average power in the time-varying bold signal in a 0.01 to 0.08 hertz power spectrum. And then we flexibly modeled associations between fluctuation amplitude and age within individual cortical regions using generalized additive models, which we basically just used to fit an age spline while controlling for sex and in-scan motion. So the first thing that we observed was that the magnitude and direction of bold fluctuation amplitude age effects varied across the cortex. We further visualized this variability by plotting fluctuation amplitude age fits in individual cortical regions. So each line in that plot is a region, and that revealed this sort of cortical continuum of developmental trajectories. And in general, sensory regions, for example, the primary visual cortex shown here, tended to show larger and earlier declines in fluctuation amplitude, which are potentially indicative of a decrease in or decorrelation of spontaneous activity. Higher order association cortices like the DLPFC, on the other hand, tended to show more inverted U-shaped curves, which were suggestive of heightened synchronized activity in early adolescence. We next tested whether developmental change in bold fluctuation amplitude was related to the maturation of intracortical myelination, which in the development literature is a very well-established plasticity limiting factor. To do this, we compared our data to results from Graham Baum and Leah Somerville's recent paper, which characterized the development of the T1-T2 ratio in youth 8 to 21 years old. And what's shown on the slide is that fluctuation amplitude and T1-T2 ratio overall age effects were very strongly inversely correlated, indicating that cortical regions that undergo larger increases in myelin content from childhood to adulthood also experience larger decreases in bold fluctuation amplitude. In addition, the age at which fluctuation amplitude began decreasing in an individual region was associated with the age at which the T1-T2 ratio maximally increased. So this association reveals temporal correspondence between fluctuation amplitude development and maturation of a main limiter of cortical plasticity. Having established that fluctuation amplitude development tightly parallel development of a plasticity regulator and broadly diverge between sensorimotor and association cortices, we sought to determine if developmental patterns spatially conformed to the SA axis. To accomplish this, I ran a PCA on regional fluctuation amplitude maturational trajectories. And the first principal component shown on the slide explained 87% of variance in development of this functional measure and can therefore be conceptualized as the principal axis of spontaneous activity development. This principal developmental axis showed striking homology to the SA axis, with principal developmental axis loadings and SA axis strengths correlating at R equals 0.7. And furthermore, if we divide the SA axis into 10 percentile bins and average regional age fits within each bin, you can see how the continuous spectrum of developmental trajectories visible at the regional level is really recapitulated by these SA axis percentile bins. Finally, we wanted to elucidate whether this sensory motor to association neurodevelopmental pattern dominated at every age between 8 and 23 years, or if it was most pronounced in a specific age range. To explore this, we quantified age-specific developmental effects at approximately 1 month intervals between 8 and 23. So here, for example, are fluctuation amplitude developmental effects at 10, 15, and 20. We then correlated these age-specific developmental effects with SA axis strength. And this approach generates age-specific correlation values that quantify the extent to which fluctuation amplitude maturational change is spatially ordered along the SA axis at a given developmental time point. And if we plot here age on the x-axis and the age-specific correlation values on the y, we can see that there is this robust correlation between developmental change and SA axis rank from 8 to 17, with peak alignment between neurodevelopment and the axis happening at 15 years of age. And then interestingly, the correlation rapidly drops to 0 near the end of adolescence, suggesting there may be this program switch in the spatial patterning of developmental processes after age 18. So altogether, this work provides evidence that neurodevelopmental processes unfold along a sensorimotor-association axis from ages 8 to 18, suggesting that molecular timing mechanisms may pattern development along this axis throughout the entire second decade of life, but not after. And given that in the development literature, prominent changes in intrinsic activity have been shown to reflect shifts in the level of circuit plasticity, the sort of cascading reductions in spontaneous fMRI activity observed along the SA axis suggest that circuit plasticity is progressively reduced along the SA axis with age. And then finally, the data I presented suggests that a main developmental axis and a prominent spatial feature axis exhibit largely convergent embedding in the brain, which signifies, I think, that spatial form in the adult brain may emerge from or be strengthened by coordinated development in the youth brain. And this idea is strongly supported by outcomes of early neurodevelopment in macaques. So during macaque brain development, there's variation in the timing and duration of neurogenesis along an anterior-posterior cortical axis. And as a result, there's also variability in pyramidal cell properties along the same axis. So regions that are more anterior have lower bouts of neurogenesis and therefore lower neuron density, but also larger pyramidal cells, excitatory cells with larger dendritic arbors that make more synaptic connections. So here, a gradient of neurogenesis length produces a microstructural feature gradient. And what this suggests for the human brain is that pattern feature heterogeneity along the SA axis may be produced or strengthened by the patterning of developmental change along this axis. And there are a couple of recent papers coming out that seem to support this idea. So the first one I'm showing here is from Adam Hines, who just became Dr. Hines a couple of days ago, just defended his thesis in Ted Satterthwaite's lab. And Adam showed that the SA axis explains continuously more variability in between network functional coupling between 8 and 23 years. And as a result, the functional coupling defined SA axis becomes more strongly embedded with age and is more pronounced, as you can see, in the brain at age 21 than at age 10. And then our friend Casey showed that a potentially related phenomenon is happening during the maturation of intracortical myelination. So during adolescence, what Casey showed is that regions that fall more in the middle of the SA axis start to exhibit myeloarchitectural profiles that look more and more like the ends of the axis. And as a result, there is this expansion of microstructural differentiation across the SA axis during adolescence. So in sum, neurodevelopment is heterochronous. During childhood and adolescence, activity-indexed developmental plasticity advances heterochronously along a sensorimotor-association cortical axis. And this temporal developmental variability may engender or enhance spatial feature variability, allowing transmodal association cortices to continuously diverge in form and function from sensorimotor cortices. And moving forward, continued study of axes of neurodevelopment may help to provide us with a more mechanistic or neurobiological understanding of gradients of brain organization. And with that, I would just like to thank the many people on this slide, including the entire Penn lab, which is led by Ted Satterthwaite, some of the collaborators not in our lab on this study, And then special shout out to Bart Larson, who's a postdoc in Ted's labs, and Ted, who were my primary advisors on this study, and just the absolute best humans to do science with.
Great. Do we have any quick questions for Valerie? Oh, yeah.
Thanks for the interesting talk. What do you think is the cognitive implication of the heterogeneous developmental gradient in the human brain organization?
The what implication?
Cognitive.
Yeah, great question. So I think that we know during late childhood and adolescence, things like executive function get a lot better in children and adolescents, improve a lot from childhood. And we also know that association cortices are developing a lot in that period. And seemingly, based on some of the data I showed and also a lot of other data, continuously diverging in their sort of neurobiological, electrochemical, structural properties from more like sensory and motor primary cortices. And so presumably the continued refinement of all of these properties is somehow supporting the emergence of like better executive function and maybe also potentially better social or emotional functioning.
Thanks.
Thank you so much. And Julien, you can come join us up here. Next, we have Julien Vezoli. Vezoli from visiting us from Frankfurt, and today he's going to talk about anatomical connectivity grading. So I'll just let you take it away. I'm pretty sure that's the
So yeah, I first would like to thank the organizer for inviting me to this very nice workshop. So the brain structure is globally organized into functional modules that integrate in even more specialized functions of the divisions. One question that arises
Okay, we have microphone on, we have a camera on, we have a presentation, we have a working computer.
So how this special anatomical and functional specialization of the brain supports such a repertoire of flexible and adaptive cognitive behavior. When we know that the brain is much more than the sum of its parts, because those parts interact in complex networks. So cognitive function include distributed neuronal networks at the local and inter-area levels. And these interaction networks are studied through brain connectomics in order to understand how the local structure connecting each specialized modules support local integration, and how long-distance connection supports higher cognitive function through global integration across modules. So we dispose of several tools to study anatomical and functional connectivity at different spatial temporal scales, and with the corresponding topological analysis in order to describe those networks in terms of aging and nodes. And this multi-scale analysis applies to anatomical and functional networks. In animals, the use of anatomical tracers with increased sensitivity led to the identification of a number of rules that govern the anatomical control. So general rules applies to, general rules applies to the rodent and non-human primate connectome, such as the exponential distance rules, which describes the exponential decrease of inter-areal connection strengths according to the wiring distance. However, some differences in the macaque connectome already point out to some primate-specific features that are highly relevant for understanding the human primate brain. For example, the ultra-high density of the mouse anatomical connector already suggests that for a given cortical area, the activity pattern is potentially influenced by widespread influence across the cortex with a high level of redundancy. So if you consider the binary dimension in the mouse graph on the right, so the presence or the absence of a link between cortical areas, there is no specificity of the connectivity profile because almost all areas are interconnected. However, this is restored when you consider connection strengths. And so in both macaque and mouse, the wide range of projection strengths, so the wide range of connection strengths across 6 orders of magnitudes, and the small inter-individual variability for repeated injections already support the notion that for each area there is a distinctive fingerprint or connectivity profiles that defines its functional specialization. So the high density and the wide range of connection strength of the cortical graphs in mammals points to the importance of quantifying projection strengths linking different cortical areas. So while anatomical connectivity is necessary for inter-area neuronal interaction, it is different. So given connection may or may not be recruited functionally depending on the context. An inter-area neuronal interaction can be studied by the entrainment and the synchronization of locally recorded signals with varying degrees. So inter-areal synchronization can be studied by phase coherence between the local field potentials of different cortical areas, and inter-areal entrainment can be assessed by non-linear causality of local field potentials from different cortical areas. So this has, and those can be spectrally resolved, so determined per frequency. This has led to the identification that neuronal rhythms in different frequency bands carries or have different neuronal interactions. So when decomposing the signal in the raw signal in slow and fast listening components, it has been shown, for example, that looking at the difference in coherence between visual pop-out versus visual search, between parietal and prefrontal areas, that gamma band coherence is enhanced in the joint bottom-up processing, and beta band coherence is enhanced during top-down processing. Later, we and others have identified that amongst the visual areas in human and non-human primates, distant frequency channels are used for hierarchical processing with a fast ethnic synchronization stronger in the forward direction and slow ethnic synchronization stronger in the backward direction. So more specifically, we directly related the patterns of rhythmic synchronization between visual areas to the non-pattern of anatomical projections that relate to a second general feature of the anatomical connector. So namely the hierarchical distance. So the team led by Henry Kennedy in Lyon made an empirical finding, which is after injecting a synaptic retrograde tracer across all the layers of cortical target area, long-distance forward projections originate exclusively from supragranular layers. And as the distance between the source area projecting to the target is shorter and shorter, there is more and more neurons projecting from supragranular layers. Conversely, for long-distance forward backward projections, these projections are originate exclusively from the supragranular layers, and for short-distance feedback projections, there is more and more projecting neurons from supragranular layers. So in other terms, it means that for short-distance projections, supragranular cells can be feedback and infragranular cells can be feed-forward. And we can come back to this later if you want. So after exhausting counting of labeled cells in different cortical areas and assignment to each area, we arrive with a density profile for each of these source area projecting to the target, and define SLN, which is the supragranular labeled neuron, and which is a ratio of the supragranular labeled cells over the sum supra plus infra labeled cells. So SLN is a continuous variable that varies between 1 for long-distance feed-forward projection and 0 for long-distance feedback projections. And for short-distance feed-forward and feedback projection, SLN tends to 0.5. So this is anatomical index of hierarchical distance. We can then order all the source areas relative to the injected area on a hierarchical scale. And combining 11 injections in 11 different areas, we could propose a model of the visual hierarchy that is highly similar to the one proposed by Penn and that demonstrates that SLN quantifies hierarchical. Now, I wanted to stress a very important point that our understanding of hierarchy as defined by SLN is much more than a simple sequential ordering of cortical areas. We have seen that SLN quantifies for each area's labeled and projecting to the target area the signature, the structural signature of the inputs to that particular local microcircuit. So the gradual changes in the variation of inputs to each cortical areas across the hierarchy defines the structural signature of hierarchy. And so we define and understand hierarchy as a connectivity gradient of inputs to each local microcircuit. I'm sorry, the animation. So and this is a link-based hierarchy. So if this connectivity gradient of input is a structural signature of hierarchy, then it is legitimate to wonder if there is a functional correlate of this. And so when we correlated this anatomical metric of hierarchical distance to a functional metric of directed functional interaction or rhythmic synchronization, it shows that in non-human primates visual areas, feedback projections synchronize more in the theta and the gamma frequency band, and the feedback projection in the beta frequency band. Now we could, using MEG, extrapolate and to human and show that gamma band synchronizations are stronger in the feed-forward direction, and beta band synchronization are stronger in the feedback direction, and allowing to construct in the same way as for the anatomical hierarchies, functional hierarchies. So those functional hierarchies are well correlated with the anatomically derived hierarchy in both human and non-human primates, but importantly, they are dynamic, contrary to the anatomical hierarchy. And we could show that some specific areas show dynamical change according to the variability context. So this hierarchical organization of rhythmic normal synchronization between visual areas of the human and non-human primates have also been observed in the human auditory cortex and between areas of the whole mental-faculty system. And these findings raise the interesting possibilities that different rhythms define distinct interaction networks, as suggested by previous analysis of power and bond ephemeral interesting human subjects. So we decided to explore the topography of large-scale functional networks and the relation to anatomical projection strengths. And for this, we exploited two unique data sets. So on one hand, the LIPRO-ECOG dataset, providing LFP from 218 sites distributed over 15 cortical areas in order to define large-scale functional interaction of two macaque monkeys performing a visual attention task. And so focusing on the period of the task with a sustained visual stimulation and top-down attention, we defined three frequency-resolved functional connectivity metric, so power correlation across trial, phase coherence, and Ranger causality. And so power correlation and coherence are undirected metric, whereas Ranger causality is a directed functional connectivity metric. So on the other hand, we used an anatomical dataset provided by the Kennedy Lab in York, providing projection strengths between 14 of the 15 cortical areas in both directions. And so we investigated whether the observed functional connectivity patterns were would be partially explained by the anatomical connectivity patterns. So first, in both monkey, we observed clear peaks in the inter-area spectrum, and with four characteristic frequency bands, so the theta, beta, high beta, and a gamma frequency band with slight variability in the frequency peak. And these peaks were also clearly identified in the power correlation for the Ranger causality spectra shown below in vanilla and red for looking. So all further analysis are focused on these four frequency bands, and we average functional connectivity across the frequency bands of interest for each monkey separately. So our first question was, do different rhythms define distinct functional connectivity network? And for this, we looked at all functional connectivity metrics between all pairs of recording sites, which represents 23,000 pairs for power correlation and coherence and double for Ranger causality. So for correlation and coherence are undirected. So on these frequency-specific matrices, we present power correlation on the upper half and coherence on the lower half. So cortical areas are ordered according, and their respective sites are ordered according to functional to anatomical hierarchy. And gray values represent non-significant points based on label computation. So we can already notice that clusters differ between frequency bands, with still some good correspondence between the two functional connectivity metrics. Now with major causality metrics, which are directed, we can see better that the 4 frequency bands define distinct interaction networks. And to further quantify this, we define distinctiveness as 1 minus R squared being the coefficient of determination across all side pairs for a given functional connectivity metric. So for major causality, distinctiveness was the least between beta and high beta network, and gamma and beta network were the most distinct. So this is really remarkable given that these networks rely on the same anatomical pair. So we then controlled for any influence of a possible different distribution of power over the different frequency bands. And we used a square root of power products. And you can see that we found the maximal distinctiveness for major causality across all frequency bands, with or without partialization according to the non-decrease of functional connectivity with intersite distance. Finally, we confirmed that functional connectivity networks are independent of power by showing maximum distinctiveness between major causality networks and power at the same extent. So next, we wanted to verify that band-specific interaction networks are modular. So we performed the modularity analysis, which we rearrange the nodes of the network in modules, in community structures, so such that highly connected nodes are clustered in the same module. And we, in order to get one consensus community structure, we combined all detected modules across frequency band, like it is usually done across subjects. So here you see one agreement matrix with 3 partitions. This is for module 1. And when we apply this to major causality network, it clearly shows that module 1 is dominated by gamma synchronization. Module 2 on the lower left is dominated by high beta synchronization. And module 3 on the upper right is dominated by beta synchronization with a high cross-module functional interaction between module 2 and 3, and little between those modules and modules. And the theta network, so lower right, is little specific. So to further investigate the point topographies of functional connectivity network, we use the strength measure, which is defined as the average functional connectivity from one side to all the other sides. And briefly, this shows that strength map display clear and meaningful topographies coherent with confirming the modularity analysis of band-specific networks. So finally, we wanted to demonstrate that weak long-distance functional connectivity deviates significantly from from randomized functional connectivity. So significant interareal functional connectivity is shown here in red for conjugate causality values, and you can see that it covers long distance for all frequencies with gamma going up to 50 millimeter, and for the other frequencies going beyond 60 millimeter. So this exponential decay with distance is reminiscent of what we have seen earlier for anatomical projection strengths. And we then investigated whether these functional connectivity patterns could be partly explained by anatomical connectivity. So we use anatomical connectivity dataset, which is providing projection strengths between all areas. So here you can see the three functional connectivity matrix with all interareal pairs ranked by anatomical connection strengths. So for each frequency band and interareal pair, functional connectivity spectral feature key have been aligned on their specific peak before average. So 14 areas were injected with tracers, and thus we sorted 91 areas for coherence and power correlation and 182 for conjugate causality. So from the left to the right, and you can see that there is a clear progressive increase in interareal functional connectivity strengths with increasing anatomical projection strength. And this is even more clear when we plot functional connectivity against anatomical projection rates. So for all frequencies, there is a significant and positive correlation, but with a wide range of correlation strengths or explained variance. So to capture the size of the FLNE effect on functional connectivity strength, we use the linear fit between log10 functional connectivity and anatomical connectivity values to estimate the expected functional connectivity effect at minimal and maximal FLNE. So because of the log10, when we use the ratio, it translates to a FLNE related fold change in functional connectivity. And so this is partly due to the, well, it's related to the regression slope, but it corrects for difference in functional connectivity that are related to FLNE, like the one drive. So when done for frequency, for all frequency bands, you can see that power correlation is the least dependent of anatomical projection strengths. Coherence is intermediate, and conjugate causality is the most FLNE dependent with so FLNE related change at the gamma frequency band, which is like tenfold. Because conjugate causality is a directed metric of functional connectivity, we decided to explore if a differential FLNE dependence would be observed for feed-forward versus feedback connections. And what we observed is that structure maximally constrained function at frequency, specific frequency channels. So the FLNE related change in functional connectivity strength is maximal at gamma, but in the feed-forward direction. Whereas the FLNE related change in conjugate causality strength at beta is only significant in the feedback direction. So you can see that the correlation in the feed-forward direction is basically high and significant. So to complete, when we then define distinct interaction networks, largely independent of the spatial distribution of power, and particularly for coherence and conjugate causality, beta, high beta, and gamma conjugate causality are largely contained in separate modules with relatively strong links between the beta and high beta module, and relatively weak links between those modules and the gamma module. Projection strength is predictive of functional connectivity strengths, but with marked difference. So power correlation is the least, intermediate for coherence, and strongest for punctual causality, and weakest for beta, and much stronger for high beta and gamma. So it's suggesting that high beta and especially gamma-based interactions prominently depends on direct cortical connections. Whether the relative independence of the beta synchronization is due to the divergent nature of feedback projections and or to the dependence to subcortical pathways, it makes beta quite a flexible frequency channel. So finally, gamma feedforward and beta feedback influences are strongly influenced by connection strengths. And while inter-area rhythmic normal synchronization can be strongly modulated by stimuli and task parameters, it is still structured by the backbone of cortical connections. So with this, I wanted to thank the co-authors and collaborators for the papers.
Is there any quick question for Julien?
What's the lowest frequency you can look at? I'm really interested in stuff below 1 hertz, but I know a lot of amplifiers that out.
Well, it depends on the sampling. Yeah, so we could reasonably go down to 2 hertz. Then after it's just. So.
Thanks. Thank you, Julien. And next up, we have Kuldeep Kumar, sorry. And he'll be talking about some genomic deletions and duplications, and hopefully some gradients too.
Hello, everyone. Welcome to the session. I stand with you lunch and you, so I will try to be brief. First of all, I want to thank all the organizers for inviting us. I still remember we had a meeting with Ernst and Casey, like with my supervisor Sebastian, we were trying to put into gradients. So I'm presenting something, and then there is also a poster presentation by one of our master students, and she has some experts on gradient also, just to give a heads up. First thing I want to give an idea is we are looking at genomic deletions and duplications. So we have genotype data, we know which people they have deletions, like instead of 2 copy or 1 copy, and then we do deletions and duplications. And then we try to map the effects of these on cognitive ability or cognition in general, we can say. And then how do these maps, like there will be some cortical maps which I will generate, that will be in the second half, and they will be like very relevant to some of the patterns which you have seen. What I want to emphasize is we do not use MRI data. There is no MRI in this. Just to, I don't want to disappoint later on. So it's like, it's no MRI. What is going on? Why did I enter in this? So briefly, like I will start with a bit of background. So copy number variants, so are deletions or duplication. So as you can see, like when you have instead of having 2 copies, you have 1 copy. So this is called a deletion. And on the right side, when you have instead of 2 copies, you have the 3 copies, it's called duplications that decrease in this case or increase the gene expression respectively. So we know that if there is instead of like having the 2 copies, you have 1 copy, so you have reduced in gene content and gene expression. And when you have instead of 2 copies, 3 copies, then you have an increase in gene content or gene expression. They are rare, that is, they are observed in less than 1% of the population, and some of the prominent ones include 22q11.2 deletion, which has been associated with increased risk for schizophrenia. So one of the important things is if we compare copy number variants with SNPs or common variants, the effect sizes that they impart like increased risk for autism or schizophrenia and multiple psychiatric disorder is many folds higher than the common variants in SNPs. The second important thing which copy number variants or genomic deletions and duplications allow us is, you know that there is a deletion, that there is a decrease in gene content, and when there is duplication, you have an increase. Whereas like if you are going to study with common variants, the directionality and like what would be the interpretation of that is still, like, at least for me, it's challenging. So in general, copy number variants, the risk, what they confer across different conditions that vary, but they seem to decrease cognitive ability persistently. So any person who will have a copy number variant might have a smaller impact on their cognitive ability or in general. To investigate this, our group and many others, they have looked into these individuals with a deletion or duplication or copy number variants. And we tried to look how does these impact the cognitive ability. And what we found is close to 50% of the coding genome, or 10,000 genes, they seem to modulate cognitive ability. That if a person has that, like a deletion of say 20 genes, that could lead to a significant decreases in cognitive ability, and that like varies even across diagnosis also. So what we don't know, like, so when we take these 10,000 genes, so that's like half of the coding genome. So, and if we try to do a functional enrichment or the Go terms enrichment, so what we observe is you have almost all of these are micro scale and cellular functions that got enriched, and it covers almost a wide spectrum of the Go terms. So then you cannot have a functional prioritization based on those. In addition, in clinic, if somebody comes and they have a deletion and duplication, then you cannot inform them more than you will have a decrease in cognitive ability. But what could be like if it's going to be some specific activities or some specific networks, that's not. So in this one, what we try to see is that the cognition that emerges likely from the interaction of these networks, and that marks the structure, like whatever interactions are occurring at the micro scale. So the brain networks, they can be seen as a macro scale version of these underlying cellular functions, and that gives rise to the cognition. So on this axis of gene, cell, network, and cognition, networks are much closer to the cognition than these genotypes. So what we are interested in our group is we know if there are deletion and duplications that leads to decrease in cognition. Can we also include some information on these brain networks and how they can inform our understanding of these impact on cognitive ability of these deletions and duplications? So we hypothesize, this is the map from the first presenter in our session, so you might have seen this. I'm showing it again. It's a really cool review, just to emphasize again from what you saw. So we hypothesize these spatial patterns of gene expression, they could capture these macro scale brain networks, and they can help us understand how these large chunk of genes that could impact a cognitive ability. So specifically what we will try to do is we will take the genes, we will try to partition them into gene sets, and then we will look into the deletions and duplications, the data which we know, and then try to come up with the effect size maps across cortex on the impact on cognitive ability when the genes are deleted or duplicated. I will try to go with the methods briefly. So in case, like if you don't follow, you feel free to email me afterwards also. So to give a brief summary, we start with the famous Human Brain Atlas, like the gene expression data. We use the vision package from Ross Muckle. And so we have the expression profiles for the genes across regions. Now for 50, we have close to 15,000 genes after processing. If we look at a specific gene as an example, like here, SST, so this is the Z-scored distribution profile of the expression, where a lighter color is like a higher expression relative, and a darker color is a relatively low expression. So what we do is to incorporate this information with the genes, as we Z-score this, we apply a threshold, and we say that anything which survives this threshold has a relatively high expression. So for SST gene, it has a relative high expression in these broad regions. So this is like a binarization you can think of, or like Now we repeat this same approach for all the 15,000 genes, and we count the genes that have a relatively high expression across cortex. So if we simply plot, like take these 15,000 genes, and if we just count based on this approach, so the mapping of the count of the genes that have a relatively high expression, it usually shows a higher value in the visual and the default mode network, as well as like somatomotor, which kind of explains that there may be like a relative high expression, and that could explain the 2 dimensions of the principal component. If we run the BCA, then these are the 2 ends which show the 2 extremes. So next is like, then comes our data. So this is not our data, Alien Human Brain Atlas, like this has been studied by thousands and thousands, if not millions of researchers. So then comes the data list. So this is a slightly unique aspect, as we have close to 265,000 subjects. We have their genotype data. We take them, we call CNV using 2 pipelines. It's QuantiSNP and NCNV. You can look at the pipeline if you're interested. We also return these CNV calls to the UK biobank to look for this, but there are some clinical data also. So you identify what are the large chunks of deletions and duplications in each individual. So a person, like say for example myself, say I have a large CNV, and then there will be some deletion of genes, or there could be some duplications, which is like instead of having 2 copies and 3 copies, I hope I don't have a CNV. That's a hope. So once we have done this, so then we know like within an individual that these specific set of genes are deleted or duplicated. So then if we look at the mapping of these genes, which we observe, so we have like across 265,000 individuals, and then we have already like generated this list of based on high expression. Then if we look in our data itself, like the genes that are deleted, where do they map? So this is the same count like the genes with relative high expression, and we are counting for each cortical region, the genes which are actually deleted and observed, and the genes that are duplicated. So these also follow the same pattern as is shown by the correlation. So it's like even if the deletion is occurring, they do not have a bias. So this was to check like, okay, if there is going to be say higher deletions in visual areas than default mode, then that could have been like there is a bias even in the observed data or deletions and duplications. So then what we did is we have for each individual, we know that they either have a deletion or duplication. We have these gene lists for each cortical region where we say this has a relatively high expression. Then what we do is for a particular individual, we count the number of genes that are deleted or duplicated that has this relative high expression. And then we also account for those which are not in this particular cortical region category. And we run a linear model for each of these 180 cortical regions from Glasser-Bar selection. So what it gives is you have a count, so you get a mean effect of a deletion. Like if somebody has larger number of genes deleted, then they will have probably like a higher score, and the mean effect, their IQ should be much lower. So what you get is these effect sizes estimates for each of these cortical regions. The main output actually is the maps which we get from these, because we know like the deletion and duplication, they are going to affect the cortical cognitive ability. So these are the cortical maps which we get, like running 180 linear regression models for deletions, like when there is a decrease in gene content, and duplications. What we find is that they show a distributed cortical distribution. Most of the color is blue, which is like there is a decrease in cognitive ability if there is a deletion or even a duplication. In general, deletions, which is like a decrease, leads to higher effect sizes. Like these are the Cohen's d effect sizes, which we are showing on linear model estimates. Then duplications, that has been shown. But what is, one slide at a time. But what is interesting is if you look at the largest effect size in case of deletions, which is they are localized into somatomotor. And if you look at the same region in duplications, you will have near zero or not so high effect sizes. And if you look at the duplications, then you have the largest effect sizes that are in resting default mode network. And if you look at the same in deletions, they have relatively lower or close to zero estimates of the effect sizes. If we z-score the same, just to give a gradient perspective to this, so if you look on the left is the z-scored effect sizes, which gives a relative ordering of these patterns. So we see that the deletions, they are impacting these broad scale networks, and these impacts, they distribute according to this gradient, and duplications also not so strongly, but they have this opposing. So if you look at the deletion and duplication map, they go on the opposing ends. If you plot these in a scatter plot, like these 180 effect sizes, like if you plot them in a scatter plot, what we observe is on the y-axis is the deletion effect sizes, and on the x-axis is the duplication effect sizes, where each point is representing a cortical RY, and they are color-coded based on the principal component of the gene expression or gene expression hierarchy. We have a negative correlation between deletions and duplications with those the somatomotor and the default motor higher order networks, grouping specifically for deletions and duplications. Sorry. And if we plot them using in a box plot, so here we are looking at the regions that are mostly sensory. So this is like we take the principal PCA and the top 30% and bottom 30%. And if you plot them as sensory and association, we see this again clearly that deletions have larger effect sizes on sensory than association regions, whereas duplication have larger effect sizes on association than sensory. Again, repeating that all the networks there is a decrease in cognitive ability across all of these maps. Third, what we did is again coming back to the maps which were presented by Valerie, like in her talk. So we take these reference maps, take our deletion and duplication effect size maps, we correlate them with the three maps which we have, and including the famous functional hierarchy. Like I have color coded this because my supervisor, he has a green, he cannot see green, so this color is probably like visible to most of the people. So what we see is the deletion maps, they show a positive correlation, and duplications, they show a negative correlation, again emphasizing opposing ends of this broader organization hierarchy may be reflected. We were able to generate these similar maps with only UK biobank, which is a general population cohort, that in general population without much clinical diagnosis also, and excluding UK biobank, which was primarily a clinical data set. So we have limited sample sizes, so that's why some of them, they are not significant. Then what we also did is we tested these correlations using that z-score thresholding, which we tried to define how relative high expression, and it's relatively stable, that even if you go up to 1%, then there would be like a smaller number of regions which will stand out. And also the differential stability, which is a measure of how stable is the gene expression for data from Allen-Human Brain Atlas across the 6 brains, and it is relatively stable. So then what we did is we looked at the replication, which is tricky because there is no other data with spatial coverage. The next closest thing which you can do is the brain span. We focused on the brain span adult data. We took the 11 cortical regions. And for deletions, we observe the similar trend. And for duplications, it's very challenging because there are only 4 points are there. But largely on the right is again the one which we generate using Allen-Human Brain Atlas, and on the left is using brain span adult data with a broad trend at least, which is similar. Finally, what we also asked is, like, our lab is more focused on this genetic severity scores and all, which has been our focus. Does that genetic severity score impacts this distribution? So we further stratify. So we cut the gene sets into 3, which is like on the bottom left is the extremely severe gene. So if somebody has these genes, ideally they will have a larger impact than the ones which are called tolerant and highly tolerant. So the effect sizes, they become larger in case of intolerant, as you can see a deeper blue color compared to these, which kind of like on the lines of what we have expected in our previous other studies using only the genetic data. But what happens is, like, if you correlate these maps again with this PC1 of Allen Human Brain Atlas, what you see is like the deletion maps are still positively correlated and duplication maps are negatively correlated. There are certain differences, but what it shows is even the genetic similarity is not changing this preference of where the impacts are distributed. And this is again highlighted in the box plot, a simplified version of the box plot, that where sensory and association, the mean effect sizes are different. So with this, what I want to conclude is like what we try to see in adults, like I want to emphasize on this one, there seems to be an optimal balance for these large-scale networks. And if you have a genomic deletion, which is like a reduced decrease in gene expression, that impacts mostly sensory, motor, and visual compared to the association. There is a broad spectrum impact. And if you have an increased gene expression, which is duplications, then there is a larger impact on these association cortices or like higher order processing regions. So this genomic balance and this hierarchy, like how and when they evolve, like I think it would be very interesting to look at some temporal aspects, like as was highlighted in the previous talks, but this is something very interesting. I would like to thank my supervisor and the collaborators. It's part of a larger CAMP project and G2MH network. And finally, shout out to more gradients and CNVs. So you can visit to the poster. So Andrea, she is going to present a poster, and then she also has a whole presentation on more CNVs and more gradients. Thank you, everyone.
Thank you so much, Kuldeep. Okay, then I'll invite the other speakers up to come take a seat. Are there any quick questions for Kuldeep? Yeah, on that.
Yeah, I was curious whether the genes that are relevant to the deletions are also very clustered in the gene coefficients from the brain atlas PC1. And I guess what you think about looking at the associations in gene space versus the spatial.
So we did look at the distribution of the genes for the say expressions in deletions. So there was a map where we tried to see if there is a difference between distribution of these genes across deletions and duplications. In general, like we do not find that difference between the deletion duplications, or if you just look at all the gene sets. So in general, there is no bias there, or like no specific, like I would say, like a preference.
Yeah, just take a seat. So now we can open up to general questions. Daniel, do you want to
take it away?
Thank you for a great presentation. I want to discuss something about the last lecture. I think it's good as also to showing the end-to-end analysis. But I think there is limitation about the explain about the causality between gene and ability. So how do you think about that?
Yeah, I think like the causality is like that's still a big question actually. So it was more like a characterization, I would say. Like we have this adult data sets, we know what are the deletions, duplications. We know like from an independent data source, like what is the gene expression profiles, and what we got is this distribution map. So it's more like characterization. Causality is maybe if I do one or two more postdocs, I will try to answer that question. But it's a very interesting question, and I think like it may be relevant also to the talk which was presented by Valerie. Like there may be some neurodevelopmental aspects, like it could be happening early in neurodevelopment. Like deletions have in general larger effects, duplications have milder effects. So that's why they may be affecting the fine-tuned, like higher cognitive networks there could be those things which could be explored, but for now I can just speculate like that. Thank you.
Hi, I have a question for Valerie. It was a really interesting talk. Thank you. And one of the graphs that you show where each line is a region and the sensory motor decrease while the high order of neuroimaging. I noticed that for earlier ages, the variability of the amplitude is larger and it becomes smaller. So how would you interpret this like change in variability across these regions?
Yeah, great question. I think probably what's happening is that earlier everything is changing more, and then as you approach like the end of the age range we looked at, like in the early 20s or late adolescence, sensory regions are stopping to develop, and so their like trajectories would flatten out towards zero, while association cortices are still changing, and maybe that explains the drop in variability.
A question for Kuldeep. You kind of talked about this balance between deletion and duplication, and how it was almost as if there was a preference is the right word, so preference for sensory for the deletion and then duplication for the association. Is there kind of balance when you're just looking at subjects that didn't have duplication or the genes that didn't have that duplication? Is there a balance between sensory and association?
So that would be kind of like, so my intuition, like our hypothesis is like all these networks, like when you have two copies, all these networks are involved in your cognition. And with the data which we have, we can only test like if there is like you, instead of two copy, there is one copy, there's an impact. And instead of two copies, you have three copy, like increased gene content, and there is an impact. But like this baseline, when you have two copies, I don't think like with this neuroimaging and gene expression data, it would be possible to test, because then we won't have like individuals with any count of the genes that are deleted or duplicated. Then we are saying that they have perfect two copies. Then every network should be contributing to the cognition, which I think is a possible thought process. But yeah, it would be difficult. It may be tested using different strategies, maybe animal models and like that, but that is not my domain, just to clarify. But that's an interesting question, actually. But we build on actually like most of the networks, like the gradients which we observe. So that's like a general population observation. So our hypothesis was what we are observing. To add on that is like, oh, this is general population, and this is deletion, this is duplication. So like that.
So you took some time to process the so I have a follow-up question. Sorry about that. So could I kind of understand it as like the maturation of low-level sensory cortex kind of being the driving force of this whole brain development? Like you said, that the reason why the variability of decreasing is due to sensory cortex. So I'm really interested in how like low-level sensory regions, the development of those level regions impact the later on development of higher cognitive functions. So can you, what are your thoughts on that?
Yeah, I don't know if the data I showed get at that necessarily. I more think it's just that because the sensory regions are maturing earlier, and like I said, like their developmental changes going to zero, and then association cortices are doing something, like that's why the variability drops. But I don't know if we can make the jump that like early development of sensory regions is what's driving something in association later. And I do think that the U-shaped curve in association cortices is maybe even more what's driving the fact that like the maturation patterns are explained by the SA axis, like that is more different, and sensory cortices are doing sort of a similar thing
I don't know if that answers your question. We can also chat after. But your question makes me think of something that I was also thinking when you were talking, which is it's sort of surprising to me that deletions have a greater effect on cognitive ability, and deletions were more expressed in sensory motor cortices, right? You might expect things affecting cognitive ability more to be more expressed in association cortex. That would be my prediction. But if we think about it from a developmental angle, like maybe if you have these deletions and your early development of sensory motor cortex is off and it's scaffolding, like the entire rest of the development trajectory, or like it's scaffolding development up this brain hierarchy, then maybe that's why it's localized in sensory motor cortex and has like a larger effect. Completely speculative, but an interesting thing to think about.
Can I go into the question? Yes. I wonder, do you believe that there's one optimal anatomical sensory association hierarchy? This goes to everyone. I'm reminded, for example, there was a paper by Klaus Gilgertag some years ago about the Feldman and Van Essen hierarchy and how you could actually reorder them in hundreds of ways. And is there one optimal ordering of the anatomical hierarchies you were talking about?
What concerns the ordering of the Feldman and Van Essen, it was classified based on qualitative principles, and we showed that with quantification, like the 7 like that I presented, you end up with a finite hierarchy. Still, it's a model fitting, so it's a global hierarchy, and you have error bars or confidence interval for each levels, which might or might not give some flexibility for each areas to jump according to the task. So I think that's what we see with the functional hierarchies also.
Yeah, thanks. Maybe a little bit towards Valerie as well. I noticed in your work, you have this really strong correlation between the developmental axis and the SA axis. The temporal parietal junction seemed to be an abnormal, it can fit with the regression line though. Do you have any suspicion why the temporal parietal junction it doesn't align between those two axes?
Great question. I don't know.
It just jumped out at you. I don't honestly, like, I think I'm more of the perspective that we need multi-dimensional characterizations of this type of organization. And so I really like the fact of quantifying Julien, but there's going to be variability across tasks as well. And I think when we start looking at like a hierarchical perspective of organization as well, rather than just one hierarchy, but multiple hierarchies either being from different sensory regions, but also in different networks, how those might be structured in different ways and how they might be activated across different tasks. So maybe as we move towards more multidimensional models, we can start understanding why some other regions stand out in onwards.
Yeah, I completely agree with the idea that to figure out, like, is there an optimal sensory motor association hierarchy, and how can we define that? I think just like looking at more data types and seeing where things converge versus diverge, and it's not clear to me if like the 10 maps that I'm showing, like, are they all capturing the same exact sort of patterning, but there's just slight variability, or are they representing like two different spatial gradients? I think, and like which ones are actually like mechanistically related versus showing similar patterns, like just by chance not causally related. I think all those questions are still left to be answered.
Yeah, Nicole.
Maybe more of a comment and still trying to put it into a question, but related to your question, if there's an optimal order, and kind of the slide you showed, Valerie, with all these different indices or descriptors of the brain that all seem to recapitulate this core or SA axis. I sometimes think it's tempting to look at them and say, oh, they're similar because they have a similar spatial pattern, but on the other hand, they are also different in many ways.
So for example, I was looking at the temporal pole in this maps, and sometimes it's at the peak of an axis, and sometimes it's intermediate, or in some of your maps, I can't remember which one, it was actually similar to a sensory map. So I think there's a lot to do still. We're not done with saying, oh, we found the principle of the brain or of the human brain. So I think there are a lot of interesting differences between those maps to uncover, and there's still work for us to do.
Yeah, I think that's a great point to highlight. And if you look at correlations among the maps, some are really strong, like 0.8 to 0.9, and then some are, 0.4. And so there's definitely more variability than might meet the eye to look at things quantitatively. And yeah, another thing that varies a lot is whether more like executive network regions are at the most association pole of like an SA axis, or whether it's DMN regions that switches a lot across the 10 maps. So yeah, definitely a lot more to figure out.
Brilliant.
Yeah, I
Brilliant. Does anybody else have a question, comment, want to share anything? I think for the sake, this has been a fantastic session. So thank our speakers again.